Hello and welcome to this lecture on seed anatomy and germination. In this unit of study, we'll be mostly looking at the seed anatomy of angiosperms, the flowering plants. There's another video about gymnosperms if you'd like to learn about the seed anatomy of gymnosperms, which differs somewhat from that of the flowering plants. Um, we'll be going over major parts of a seed and how they function and develop. And we will also be talking about the germination process. To begin with, what are seeds? Seeds are the reproductive unit of the seed plants. That's the clade spermatophyta. Not all plants produce seeds, but the plants that do produce seeds, they are the spermatophytes. And the seeds develop from the fertilized ovule within the ovary of a flower. Seeds are composed of an embryo or a baby plant, a food source, and a protective covering. And germination is the process of the seed sprouting and beginning growth. So those will be the main focuses of today's discussion. Angiosperms possess a seed coat. Their seeds, sorry, I should say the seeds of angiosperms possess a seed coat, which is a protective layer covering the seed. Endosperm, which is the food stored in the seed, and the embryo, which is the baby plant. Um, in these two diagrams, we can see a corn seed, which is a monocot, and bean seeds, which is an example of a dicot seed. Monocots and dicots are two of the main groups of angiosperms, and their seeds differ in some significant ways that we will discuss as we move forward. We're going to start from the outside and move in as we start our exploration of seeds. So we're going to start with the seed coat. The seed coat is also called the testa, and the function in very loose general in a very loose general sense is to protect the internal parts of the seed. Um, the reason I say in a loose general sense is because not in all species does it really do that. A good example of a very, very protective seed coat would be the seed coat of an American lotus. These black seeds that you see in the top left image are American lotus seeds, and they have an extremely hard seed coat. It takes a minute with sandpaper to get through that. Um, Corn may look like it has this bright yellow seed coat, but that thin yellow layer on the outside is actually the fruit, and the fruit layer is tightly adhered to the underlying seed coat such that they're inseparable. Um, these black-eyed peas in the bottom right corner are a species of bean, and they have a very, very hard seed coat. But the closely related peas, sorry, not peas, but peanuts, you'll see that they have this thin, thin, papery seed coat. And the reason why peanuts have a thin, papery seed coat is rather than using the seed coat for protection, peanuts have evolved to use their fruit for protection, the shell of the pod that the peanuts are contained within, which has meant that there's no selective pressure toward having a thick protective seed coat. So they have a thin, papery one. So all seeds have a seed coat, but it can vary a lot between different species, both in its color, composition, structure, form, hardness, etc. Um, the seed coat has a couple features that are important to look at. The hilum is the first. The hilum is the scar where the funiculus was attached and through which the developing seed received nourishment from the parent sporophyte. So the parent sporophyte, that's just the parent plant that produced the seed. Um, and the funiculus is essentially like the umbilical cord of the seed that supplies nourishment to the developing seed after fertilization. Because we often compare the funiculus to kind of like an umbilical cord, we could compare the hilum to maybe the belly button of a seed. Um, just under the hilum, we often have a structure called a micropyle, and the micropyle is an opening in the ovule through which the pollen tube delivered sperm to the egg. So just to review the process of pollination and fertilization, the pollen grain has to land on the stigma of the pistil. The pollen grain has within it a tube cell, which grows down toward the ovules through the style into the ovary, and it's following chemical cues which are released by the ovule. The generative cell then travels down that tube. It divides into two different sperm, and those sperm then are able to enter the ovule through the micropyle and fertilize the egg. Seed coats often have additional appendages. One of them that you see with some frequency are arils. Arils are an expanded funiculus which engulfs the seed coat in a soft flesh, and that's often used to attract seed dispersers. The example I provided in these top two images are magnolias. Remember, the funiculus is like the umbilical cord that supplies nourishment to the developing seed. So in this case, the funiculus, rather than just attaching at the hilum, is actually expanded and kind of wraps around the entire seed with this fleshy layer. And it's a way of getting your seeds around. So if we look at this magnolia in these top two images, when we look at the fruit of a magnolia, it's an aggregate of follicles. So each follicle is a fruit that splits open along just one seam, and it contains one seed. So when each follicle splits open, it releases the one seed that was contained within, and we can see that those seeds have this red fleshy layer covering the outside of the seed. But then we see in the top right image how when we strip off that aril, we still have a seed coat which exhibits a hilum and a micropyle. So that red fleshy layer is not the seed coat. It's an additional layer produced by the expansion of the funiculus, kind of wrapping the seed coat up in a bag of flesh. Um, and you'll notice that these arils on magnolias are bright red. Birds are often attracted to the color red because birds have excellent color vision. And if you have green leaves, red is the highest possible contrast because it's a complementary color. So red stands out extraordinarily well against a green background to birds, which makes those arils on the seeds very easy to find, and convinces birds that it's a good thing to eat. They'll eat the seeds and then defecate those seeds somewhere else. Um, and as always, it's always an advantage to have your seeds eaten by an animal because the animal's digestive tract contains strong acids which can disinfect the seed. And then when the seed is deposited, it's deposited with a little bit of fertilizer in that manure or dung that the animal is dropping the seed in, so gets seeds off to their best start. An elaiosome is a fleshy seed appendage containing amino acids and lipids for attracting insects such as ants to disperse seeds. So amino acids are the precursors to proteins, and lipids are just fatty molecules like oils, fats, waxes. And ants find these irresistible. A lot of the ant species that disperse seeds possessing elaisomes are species of ants which are specialized to scavenge the bodies of dead insects. And so often the eliasome um, contains compounds in it that these ants are smelling for and searching for in their normal food. And having ants drag you away has several advantages. Ants are really only interested in that fleshy portion. So typically the ants will drag the seed with the eliasome back to their colony. They'll chew off the eliasome and then they will throw the seed away in their rubbish pile. And often their rubbish pile of an ant colony has a lot of decomposing material in it that provides extra nutrients. And ants also have big problems with fungus, and so ants will actually secrete antifungal compounds that can help prevent the death of those seeds during germination. Um, and then the last type of seed coat appendage we'll be discussing today is the sarcotesta. So we just discussed how testa is a synonym of the seed coat. So a sarcotesta, sarco means flesh or muscle. Um, so sarco is a fleshy seed coat or testa. And that's a fleshy seed coat used to attract seed dispersers. Probably the most familiar example would be a pomegranate. Every seed in a pomegranate has this fleshy layer on the outside that is actually the seed coat. And if you've ever eaten a pomegranate or had to cut one up, you'll know that the actual flesh of the fruit of the pomegranate is very, very dry and spongy and pithy. And that doesn't make for a delicious meal. So wild pomegranates actually burst open when they're fully ripe, exposing the seeds and their fleshy seed coats to birds. And so birds will show up and they can pluck the seeds off. The seed coat is digestible, um, but the remnants of the seed inside are perfectly good at germinating all on their own. They don't actually need to have that fleshy seed coat in order to germinate successfully. So that's kind of an interesting feature. And pomegranate breeders specifically select against the bursting trait because you can imagine that that would greatly reduce the value of your pomegranates at the store if they were completely busted open. So um, most of our modern pomegranate varieties are selected against that bursting behavior that we see in wild pomegranates. Endosperm is the food stored inside the seed to fuel germination. There's a frequent misconception that seeds are living off of food reserves while they're waiting to germinate, and that's not the case. Dry seeds are metabolically inactive. They're not really doing anything. They're just kind of in a state of stasis. The endosperm or the food is stored in order to power the germination process, which we will be exploring more in depth later. Endosperm, being food, is mostly composed of things that we would recognize as food: proteins, carbohydrates, and lipids or fats and oils. Um, Proteins are super important for building cellular components. Proteins are like little machines that do all the work within the cells. So having some protein in your seed is really helpful for getting a head start on building cells. Carbohydrates aren't a major component of most cells themselves, but they are a very easy to process energy source, which can give you some quick energy during the germination process. And lipids, which are oils and fats, you'll notice that rather than containing 4 kilocalories per gram, they contain 9 kilocalories per gram. So they can contain over double the amount of energy per gram of seed weight. So seeds are capable of storing really large amounts of energy within a relatively small volume of seed. And those lipids aren't just useful as a dense energy source, they're also valuable for building cell components like cell membranes, which are made of lipids. Um, and the endosperm composition varies by species. So when we look at these different seeds here, we should be able to recognize all these seeds as popular foods for human beings because we like to eat seeds for the same reason that the seeds like to, store store food. I mean, it's food, right? So when we look at these almonds, for example, almonds are primarily protein and fat. They contain primarily proteins and lipids. Wheat contains primarily carbohydrates and some protein, usually around 11%. And then peas are mostly protein and carbohydrates. So there are some variations in um, which nutrients we have based on the species. And I have got here this discussion question: why might seeds vary the composition of their endosperm? I'm going to give you a second to think about it before we come back and discuss it. Why might seeds vary the composition of their endosperm? The example that I like to use to illustrate this point are poppy seeds. Poppy seeds are famously very, very, very tiny. Um, and they also contain a fair amount of lipids or oil. If you're going to have a really, really tiny, tiny, tiny seed, it makes sense to store as much energy in the densest format possible within that seed in order to guarantee that it has energy for to germinate. So sometimes the composition of the seed um, varies because of seed size. Sometimes it's going to vary um, because a plant is trying to dissuade certain animals from eating its seeds. Sometimes there are environmental challenges. For example, um, oaks and avocados and many trees which live in forests have very, very large seeds in order to give their germinating seedlings the greatest chance of survival because there's so much food stored in a large acorn or an avocado pit that the seedling is able to grow, a really big root and a shoot and still be receiving nutrition from that original endosperm, potentially years after it's germinated, which helps it get established in the dark shade of the forest where there's not a lot of light, and so it's hard to do photosynthesis and make your own food as a young tree. Double fertilization is something worth understanding, especially when we look at seed development and um, successful seed production. Um, Double fertilization only occurs in the angiosperms or the flowering plants. It's a unique feature wherein one sperm cell from the pollen grain fertilizes the egg cell, and the other sperm, which is produced by the same pollen grain, fertilizes a specialized cell, which is called the central cell of the embryo sac. Um, and those that cell contains what are called the polar nuclei. Um, and because we've got two nuclei plus the sperm, we end up having three copies of each chromosome, which we would describe that as triploid. Human beings are diploid. Di means two, and then ploidy is the number of chromosomes, number of copies of each chromosome an organism has. And because those polar nuclei are together in the same cell and then fertilized by the sperm, having three copies of each chromosome, they can't develop into a second embryo, right? Or baby plant. The egg, when it's fertilized by the first sperm, is going to develop into an embryo or a baby plant. But the polar nuclei are going to follow a different developmental path. They are going to, after fertilization, develop into the endosperm. They're going to divide and grow larger and become the stored food inside of the seed. So just to review, the polar nuclei are two nuclei contained within the central cell of the embryo sac. Um, After fertilization, they develop to become the endosperm or stored food. Endosperm can develop in a couple different ways. I hope we're all familiar with cell division, mitosis. There are ways to do cell division that are a little bit, I suppose, interesting that don't involve the creation of actual new cells. Um, in our first type of endosperm development, we can actually have cell nucleus divisions, creating basically new nuclei without creating any new cells. So as more and more nutrition enters the seed through the hilum, through the funiculus, um, our nuclei divide and multiply, their DNA, and they produce more endosperm, but they don't create any new cell walls. So there aren't actually individual cells. There's basically just one giant cell containing a huge number of nuclei. And a good example of nuclear endosperm would be coconut water. So if you've ever cracked open a coconut and spilled out the coconut water inside, That's an example of nuclear endosperm. Because it's all one kind of giant cell, it it's liquidy. Cellular endosperm is when we have full, complete, normal cell division creating the endosperm. So we do form cell walls. And a good example of that would be the coconut meat within a coconut, which tends to be more solid because of the presence of cell walls. And then halobial endosperm is basically a combination of both methods within the same seed. So the first um, endosperm cell, which is formed by the fertilization of the polar nuclei, divides in two, and it basically forms two parent cells. One of those cells is going to go on to perform nuclear endosperm division, and the other is going to go on to perform cellular endosperm division, which is going to result in a seed which contains a combination of cellular and nuclear endosperm. So a coconut is a good example of a species that has halobial endosperm with both kind of solid cellular endosperm and liquid nuclear endosperm. Most monocots do produce halobial endosperm, but we see a lot of variation across the plant kingdom in exactly how that endosperm develops. The endosperm or the food within the seed can also be stored in a couple different ways. So far we've been discussing the endosperm as a unique part of the plant. Um, a seed is essentially a baby in a box with a lunch. The baby being the embryo, the box being the seed coat, and the lunch being the endosperm. But sometimes the baby eats its lunch before germination, and sometimes it keeps its lunch outside of itself until it germinates. So, in our first food storage type, endospermous seeds or albuminous seeds, they store their food in the endosperm in that fully developed seed. So, if we look at this diagram at the top, we can see that um, in the top right corner, we have a dicot, an endospermous dicot seed, right? And we can see the cotyledons, the epicotyl, hypocotyl, and radicle, which are all parts of the embryo. And then outside of the embryo, we see the endosperm labeled. So we've got an embryo surrounded by endosperm contained within the seed coat. And that's an albuminous or endospermous seed, meaning that the food is stored outside of the embryo. In some species, however, all of the food in the endosperm is absorbed into the embryo before germination occurs. And we refer to this pattern as, we would say that they're exalbuminous seeds or cotylospermous seeds. Um, and often what they're doing is they're absorbing all of the food into the embryo, especially the cotyledons or the seed leaves. The cotyledons are the first leaves that open up and expand and do photosynthesis. If you've ever grown a vegetable garden or maybe a flower garden and you've planted seeds out in your garden, they'll often come up with two little leaves that are a different shape than the mature leaves of the plant. And those are the cotyledons. In a bean, for example, which is kind of our middle top image, where we've got a cotylospermous dicot, we see that the cotyledons or those seed leaves have absorbed all of the food storage. So it's actually already ready to go inside of the embryo rather than outside. We also have some seeds which have an intermediate food storage type where they're storing some food outside the embryo in the endosperm, and they're storing some food inside the embryo. So this bottom image, I don't know what species of plant this is, but we can see that within the seed we have these very, very large cotyledons or seed leaves. And we also, which indicates the size of that would indicate to me that they probably already contain a decent amount of stored food. But we can see that there's also still some stored food within the seed coat outside of the embryo. or the baby plant. So plants can kind of exist anywhere along a range of having all their food outside of the embryo or having already consumed all the food and storing it inside of their embryo or baby plant. Um, a unique group of angiosperms or flowering plants that have really unique seed anatomy are the orchids. Orchid seeds actually entirely lack an endosperm. So if you were to collect orchid seeds and scatter them on some potting mix and expect them to germinate, you would fail miserably because orchid seeds don't have any stored food to power the sprouting process. In the wild, most species of orchids have symbiotic relationships with particular species of fungi, and those fungi respond to chemical signals in the orchid seeds, and they will actually feed the orchid seeds in order to power their germination. Once the orchid has germinated and has leaves, the orchid is capable of performing photosynthesis, and the product of photosynthesis is sugar. So the orchid is actually able to feed sugars produced by photosynthesis back to the fungus. So in the long term, the fungus benefits by helping the orchid seeds germinate because it's going to get a treat of sugar in reward in the long run. But that's a very unusual strategy and certainly restricts where certain species of orchids can grow because often a species of orchid may only associate with one or two species of fungi, which can help them germinate. The embryo is the baby plant inside the seed. The embryo develops from the fertilized egg within the ovule, and they're composed of a couple parts, the radicle, the hypocotyl, and cotyledons, and then some seeds that are extra well developed already have an epicotyl and a plumule, and we'll discuss more what those are in a minute. But I'd like to direct your attention to this left-hand diagram. We can see the seed coat in this diagram, we can see the stored food or endosperm, and we can see the embryo. And what I want you to understand about this diagram of an embryo is that all of the major organ systems of the plant are already present at this stage. The bottom nub, where it says radicle, that is the embryonic root. Then the cotyledons, those are the first leaves, those are the embryonic leaves. And then the little round bud between the two cotyledons, that's the apical meristem, which is basically the growing point or the bud from which a stem will grow. So we already have a stem bud, some leaves, and a root inside the seed. And that will allow this embryo to kind of rapidly develop from just an embryo into a plant once it begins to germinate. The first part of the embryo that we'll discuss, and arguably one of the most important parts, is the radicle. The radicle is the embryonic root of the seed. And during germination, that radicle bursts through. Well, first the seed has to absorb some water. Once it's absorbed some water, then the radicle is able to burst through the seed coat, and it's going to start to absorb water and mineral nutrients from our substrate. That could be soil, that could be potting mix. Um, the radicle is notable because it's almost always the very first part that begins to develop and grow, usually before all other parts of the embryo. Why might it be important that the radicle erupt from the seed coat and begin to absorb water and mineral nutrients before the other parts of the seed develop? I'm going to give you a second to think about that, and then we'll discuss it. So if you thought it's important for the radicle to develop so that we have the water to continue to inflate and grow those other embryo parts, you're correct. Without access to water, plants can't grow. The process of growth is essentially a process wherein plants make new cells and then inflate those baby cells with water to make them larger. So if you don't have a reliable source of water, it doesn't matter how many cells you've made, you can't inflate them with water and you can't make them larger. Um, so the radicle almost always has to develop first before the germination process can continue and proceed. The next part of the embryo that I'd like to discuss are the cotyledons. I've switched to showing you an image of a germinated seedling just so you can really get a clear idea of what these cotyledons maybe look like. Um, the cotyledons are the embryonic leaves of a seed, and in species which are um, cotylospermous, or that store their food inside of their cotyledons, the cotyledons are often quite large and often compose the majority of the volume of the seed, like in beans. Um, in species which are endospermous, we might see slightly smaller cotyledons. The cotyledons typically have one of the following functions. They're either storing food within the seeds, as is the case with cotylospermous seeds, or they're performing the first photosynthesis after germination. So once the seed sprouts, those cotyledons um, spread out, they expand, and they're green in many cases, and they're able to do photosynthesis and help the plant produce its first true stem. Um, when we look at the cotyledons are a very important feature of the seed, also for taxonomic reasons. There are two main groups within the flowering plants, the monocots and the dicots. The dicots are flowering plants that have two cotyledons per seed. Good examples would be like oaks or tomatoes or beans. And when those species germinate, or if you were to dissect their seeds, you would find two cotyledons. So looking at this germinated seed on the left, we can see, I think this is a squash seedling or maybe a sunflower. We can see that there are two large seed leaves, and then we can see some new baby leaves that are the true leaves developing at that bud where the stem tissue is going to develop. Monocots are flowering plants that have only one cotyledon per seed, and that would include things like grasses, orchids, lilies, palms, etc. Um, the monocots have only one cotyledon per seed, and so you only get one seed leaf rather than two. So that's an interesting feature. It's something to look for. Very valuable to be able to identify monocots versus dicots. Monocots and dicots do differ in other ways. For example, monocots tend to have parallel veins, dicots tend to have branched veins. Monocots tend to have floral parts and multiples of threes and fives. Dicots tend to have floral parts and multiples of um, fours and fives. So there's some other differences as well, but these are kind of the two major groups of flowering plants. Um, the hypocotyl is an embryonic stalk below the cotyledons. Hypo means below, cotyl means cotyledons. You can think about hypothermia is when your body temperature, or thermia is below or hypo, where it's supposed to be. And the hypocotyl in some seeds functions to push the cotyledons above ground to begin the process of photosynthesis. The epicotyl is not present in all seeds. The epicotyl is the embryonic stalk above the cotyledons. Some species of plants wait to produce an epicotyl until after they have germinated, and they basically just have a little bud inside the seed rather than having a preformed epicotyl. Okay, and epi means above, cotyl is a reference to cotyledons. You could think about epinephrine, which is the same thing as adrenaline. Epi means on top of. Um, Nephron is Greek for kidney. So epinephron means on top of your kidney because that's where your adrenal glands are that produce the hormone adrenaline or epinephrine. So epi is above, cotyl is cotyledon. The plumule, like the epicotyl, is not present in all seeds, But where it is present, the plumule is a pre-developed set of true leaves inside the seeds, and it will become the first true leaves the new plant will produce. Beans have a pretty well-developed plumule that you can actually see with the naked eye if you dissect a bean. And that comes from the Latin word plumula, which means a little feather. The embryos of many species do not possess a plumule while they are a seed. A really good example are sunflowers. Sunflowers do not have an apicotyl or a plumule above the cotyledons. They simply have a radical, cotyledons, and a little bud that will develop into the epicotyl or the um, kind of first mature shoot. Um, they don't they don't bother developing an epicotyl or a plumule until after they have already germinated. Beans, however, have a really well developed plumule and epicotyl. So when we look at this image on the bottom, this diagram, kind of diagramming germination, we can see that we start out with a seed. In number one, it's been planted, it absorbs some water. The radical or the embryonic root erupts through the seed coat in number two. In number three, the radical continues to expand and the hypocotyl begins to develop and lifts the cotyledons up out of the soil, removing the seed coat. The cotyledons then expand, which are those seed leaves, and then the epicotyl and the plumule begin to expand. And we can go from seed all the way to a developing plant over the sequence of 5 steps. The final step within the development of a seed, we've got to develop um, the endosperm, we've got to develop a seed coat, we've got to develop an embryo. The final step is desiccation. And desiccation is the process of a seed drying out, often to less than 10% moisture. That might sound like it's pretty moist, but consider that a dry sheet of paper on your desk has between 5 and 7% moisture. If you are living in a really, really humid climate, like, I don't know, Seattle, your paper could have up to 10% moisture in it on a really humid day. So the contents of the seed are really extraordinarily dry, about as dry as a sheet of paper. Um, and The process of drying out is often controlled by the mother plant. The mother plant is providing moisture and nutrients to the seed through the funiculus, and the mother seed can kind of slowly wean the seedling off of moisture and allow it to dry down. This isn't the case in all seeds, but it is the case in many. And This desiccation process is notable in seeds because for most organisms, we know that desiccation results in death. Human bodies are between 60 and 70% water. And if we were to dry out to just 10% moisture, we would be very, 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 very dead. Um, and the reason why cells die when they dry out is because of a collapse of cellular structures and a change in the chemical environment within the cell. So you can imagine plant cells are contained within a cell wall. As that plant cell dries out, the cell membrane within the cell wall can tear away from the cell wall. Things like the nucleus of the cell could collapse. The collapse of the nucleus could um, break and fragment the DNA contained within the cell. All of the other organelles are going to crumple and their membranes are going to tear. And basically, as the cell dries up and shrivels and shrinks, it's just going to get shredded apart and destroyed as it folds in on itself. To prevent this process from occurring, the cells within the embryo in a seed pack all of their organelles. If it's been a while since you've taken any kind of cell biology class, the organelles are the little organs within a cell that make it function. And the cytoplasm is basically the gel or the liquid within which the organelles are floating, as it were. Um, the cell packs its organelles and cytoplasm with specialized proteins and sugars, and by packing as much of these protective proteins and sugars as they can within the contents of the cell, they're able to stabilize and preserve the shape and structure of those cell components as they dry out. Um, they're literally making a glass within the seed. So, in order to understand glass, I'm going to refer a minute to the glass that we're most familiar with, which is made of silicon dioxide. So, under normal conditions, or I should say, under slow cooling conditions, so melted silicon dioxide will slowly um, solidify out of a solution, or out of a melted form. And if it has a really long time to cool, um, it will form a crystal. And we know silicon dioxide crystals as quartz. And crystals have a ordered, predictable, rigid lattice structure. All crystals have a predictable, repeating structure. So if we look at this top diagram, we can see that we've got these hexagonal lattices between molecules in a crystal. In liquids, the associations between different molecules are disordered, and because everything's moving around. Um, and so the relationships between different molecules, the chemical relationships between different molecules within that liquid, are constantly changing and disordered. However, if you rapidly cool a, um, a liquid with, or maybe a melted um, crystal, for example, silicon dioxide, um, and then you rapidly cool it, what ends up happening is that liquid does not have time to organize itself into a crystal lattice. And so it ends up getting frozen in place as a solid, but with a disorganized structure. So when we think about quartz, for example, quartz is silicon dioxide, and it's extremely hard and resilient because it's a crystal. Um, Quartz is one of the hardest minerals that exists because it has this structure. Glass is the same mineral. It's not the same mineral. It's the same chemical as quartz, but it does not have the same structure. So if you take a piece of quartz and scrape a piece of glass with it, you will scratch the glass, but you will not scratch the quartz. Um, so basically, the cell is packing in a bunch of proteins and sugars and creating this disorganized but stable solid to preserve all of its organelles. It's basically just stuffing the contents of the cell with proteins and sugars so that it can survive drying out without dying. Um, and just another opportunity to dispel the myth that seeds are consuming food while they're in the dry state. If they're totally dried out, they're inactive. Chemical reactions in cells don't occur well without water. So the desiccated seeds, they're alive, but they're entirely inactive. They're not consuming their stored food. That stored food will be used when the process of germination occurs. Um, Sowing is the process of planting a seed. So at this point, we're transitioning away from the parts of the seeds, and we're really starting to look more in depth at the germination process. Um, and Where we start in horticulture with germination is normally when we sow the seed or plant it. Germination is the process of seed sprouting and initiating growth. And we've got really specific conditions that are required for that process to occur. We have to have water, we have to have the right temperature, we have to have oxygen, and we have to have the proper sowing depth. Once again, you can refer to this seed germination diagram. The seed first absorbs water. The radicle or embryonic root erupts from the seed coat. It expands and begins to absorb water and mineral nutrients from the substrate. As it absorbs water, that allows the other components to inflate with water and expand. Um, the cotyledons open up in some species, and then we can begin shoot growth from that little bud between the cotyledons. So the first stage of germination um, requires water, and this process is called imbibition or imbibing, and that's the absorption of water into the seed. One of the unique or interesting things about seeds is, um, unlike mature um, growing plants, seeds have tiny, tiny cells which are packed full of goodies. Whereas mature growing plants have large expanded cells which have a lot of water in them. By having these really tight, compact little cells which are stuffed full of food, seeds are able to imbibe water and then inflate those preformed cells, which allows them to very rapidly expand and begin growth. Looking at these images, we have some dry peas that have soaked in water for 24 hours, and you can see that in 24 hours, the size of the seed doubles. Imbibition is our first step, and we need to have water. Um, Plants are most susceptible to dying from dehydration when they are germinating. If we were to take these partially germinated peas where we've got them inflated with water and then we allowed them to dry out completely again, that would probably kill them. So it's really, really important to keep your seedlings wet throughout the germination process. Additionally, we have to have optimal temperatures for germination to occur. Your optimal temperatures that are required for germination are going to vary by species. Some species like it to be very, very hot for germinating, warmer than 80 degrees. Some species are going to prefer much cooler temperatures, maybe in the 50s or 60s for germination, and that will be their preferred temperature. So it's important to do a little bit of referencing with scientific resources and figure out what the optimal germination temperature is for the species that you're attempting to germinate. However, as a rule, warmer temperatures are usually going to speed up your germination process. Warmer conditions also favor your seed against fungal soil pathogens uh, like Alternaria or Phytophthora or Rhizoctonia, which are all fungal pathogens which can attack seedlings. Um, in some species, however, if the soil is too warm, for example, in lettuce, if the soil temperature is above 80 degrees Fahrenheit, you will see inhibited germination. The seeds won't necessarily die, but the heat will make them go dormant. And make them resistant to germination. So it's important, like I say, to make sure that you're warm, but maybe still within the temperature range that's preferred by that species. Cooler temperatures are typically going to reduce the speed of germination. Um, they're going to retard the germination process. And unfortunately, if you have really cool, moist conditions, fungus really like kind of cool, damp conditions. So often your seeds, as they're germinating slower, they're not as resistant to, they're not as vigorous, they're not going to be as resistant to pathogens, and you can have a greater percentage of seedling loss to fungus. Oxygen is also necessary for the germination process because it's a necessary ingredient for cellular respiration. If we consult this diagram, we can see that photosynthesis is the reverse reaction of cellular respiration. In photosynthesis, we're capturing carbon dioxide from the atmosphere and we are hydrating that carbon using water from the soil to create a carbohydrate sugar, right? In this case, glucose. And in cellular respiration, oh, sorry, I just wanted to add that in photosynthesis, oxygen is a waste product. But in cellular respiration, we're consuming glucose or sugar, we're reacting with oxygen, we're oxidizing it, and then we're releasing water and carbon dioxide as waste products. Photosynthesis is essentially about capturing energy, and cellular respiration is about releasing stored energy. So seeds contain a large amount of stored chemical energy, and we can't release that stored chemical energy without performing cellular respiration. So the cellular respiration process is essentially the process by which an organism releases energy from a food source. And a kind of simplified equation of that would be C6H12O6, which is the chemical formula of glucose, plus 6 oxygen molecules, 6O2. We're going to see the release of energy. And then as waste products, we're going to produce 6 molecules of carbon dioxide and 6 molecules of water. And if we turn that reaction around and we add water and carbon dioxide and we put in some light energy in photosynthesis, we're going to produce oxygen as a waste product and glucose or sugar as a product. So photosynthesis and cellular respiration are just reverse chemical processes. Photosynthesis is done within the chloroplast inside the cell. Cellular respiration is done inside the mitochondria. Without oxygen, seeds can't break down the stored energy that they have either in their endosperm or sometimes stored inside the embryo to power the germination process. Um, in a practical sense, this means that we want to make sure that we're not trying to germinate seeds in anaerobic conditions where there is no oxygen. So using a nice light, fluffy potting media that has some air spaces in it is beneficial for germination. It's also beneficial for the developing roots because they're less likely to experience root decay if we have some air spaces in the soil or potting media that our seeds are planted in. Optimal sowing depth is also beneficial for germination. You're going to have much better outcomes if you sow your seeds to the correct depth. And once again, this is going to vary by species. As a general rule, no deeper than 2 to 3 times the height of the seed is a good recommendation. It's kind of a good rule of thumb. But this certainly does not apply to all seeds. Really tiny seeds, for example, poppy seeds, if they don't have any light, if they're not exposed to light, they will remain dormant and they will not germinate. So a lot of seeds actually have to be sown just directly on the soil surface and then just kept moist until they germinate, and they'll germinate just fine on the soil surface if they're really tiny. I basically look at a seed and say, if this seed is so small that it's hard for me to physically handle with my fingers, it probably needs to be surface sown. If it's basically just a little speck of dust, it's probably a surface sown seed. We can see in the top image someone planting chickpeas to the correct depth. You can often kind of compare the diameter of the seed to your fingertip and figure out, okay, to which knuckle or what part of my finger, what wrinkle on my finger would get me 2 to 3 times the depth of that seed. A chickpea is maybe about the height of your fingernail. So if you go up to your first knuckle, pushing it down, that's probably adequate planting depth. In the bottom image, we can see a germination depth trial on wheat. And we see that when wheat is germinated 2 to 3 times the depth of the seed, we get the greatest quality and growth from our germination. The seed is able to use relatively little energy to get to the soil surface, and then it's able to dedicate a lot of energy stored in the seed towards the growth of multiple new shoots. So our first plant towards the left is the largest and healthiest and has the greatest number of tillers or new stems. Our second seed is planted about 6 times the depth of the seed, and we can see that we've got significantly poorer quality growth. The seed had to expend a lot more energy to get to the soil surface and start getting light for photosynthesis, which has put it behind. Our third seed on the right has used up almost all of its energy getting to the soil surface, which has meant that now the resulting plant has no seed resources to help jumpstart its growth. And so we have significantly poorer growth than either of the two other plants. The plant's kind of spindly and weak and not doing great. So just by sowing at the correct depth, you can really get a huge improvement in resulting plant quality. There are 2 different patterns of germination that we see, and I think that they're valuable to discuss. In epigeal germination, the cotyledons are pushed above the ground, and often their function there is to do the first photosynthesis for the plant. So in beans, for example, we can see that the cotyledons are pushed above ground during the germination process, and they're pushed above ground by the growth of the hypocotyl, which is the embryonic stalk below the cotyledons. Not all species germinate in this way. Some species have decided that because they have their food stored in the cotyledons, it's too risky to push the cotyledons above ground, because if they do push their cotyledons above ground, there's a greater likelihood that maybe they'll be eaten by maybe a bird or a rodent. So they actually keep their cotyledons below the ground during the germination process. The hypocotyl is relatively short, and they're instead using their epicotyl to push a shoot above the ground. So in epigeal germination, the cotyledons are pushed above ground. In hypogeal germination, they remain below. Peas are a good example of a species with hypogeal germination. Dormancy is a bit of an interesting subject within horticulture. A lot of people really hate dormancy because it can make seeds really, really challenging to germinate. Dormancy is a state where your seed is alive, but it's physiologically incapable of germinating even under ideal conditions. So even if you collect the seed and give it the right amount of water, the right amount of oxygen, the right temperature, and the right sowing depth, it will sit there inactive in the soil, which can be really, really frustrating as a horticulturist. So it's important to understand that plants have evolved dormancy mechanisms as survival strategies. And once we understand those survival strategies, we're able to then manipulate seeds in such a way that we're able to induce them to germinate by overcoming dormancy. Dormancy helps plants survive in many ways. One of the first and most obvious is the prevention of precocious germination. So if we look at this top image of a tomato, um, that's not supposed to happen. The seeds are not supposed to germinate while they're inside the fruit. And normally, seeds have dormancy mechanisms that mean they have to desiccate or dry out first in tomatoes before they're capable of germinating. But in this case, in that moist, warm environment inside the tomato, they've decided there's optimal germination conditions and they've sprouted. If this tomato were eaten by an animal, all of its embryos would be digested and die. Whereas if the seeds were adequately dormant, they would be eaten by that animal. The seeds would pass through the digestive tract unharmed, and then the seeds would be able to germinate in the dung of the animal. Dormancy is super important for preventing precocious germination. It's also important for ensuring seeds germinate at the optimal time of year. If you are a plant which sheds your seeds in the fall, you don't want your seedlings to germinate in the fall because they'll get killed immediately by the winter cold. You might want to have some chemical mechanisms in your seed that make sure that the seed does not germinate until it's gone through a period of cold called a stratification, because that will ensure that your seeds don't germinate in the fall, they germinate next spring, when conditions are going to be more optimal for seedling survival. Dormancy can also ensure sporadic germination to survive adverse events or to help seeds survive long periods of time in unfavorable conditions. Um, this plant in the bottom is an American lotus, and we see again those large black seeds of the American lotus. It's this plant with the large leaves and the yellow flowers. American lotuses have a bit of an interesting history. They have a really large native range stretching from eastern Canada all the way down through the Midwest and eastern United States down into Texas and northern Mexico. And in the northern part of their range, the American lotus was actually nearly eradicated um, due to some of the consequences of fur trapping. So, French and American fur trappers were trapping animals like otters and bears and foxes and coyotes and everything. And the trapping of all these large predators, especially aquatic ones, led to an explosion in the muskrat population. And muskrats feed primarily on aquatic vegetation. So if you have a huge muskrat population, they're going to eat all of the American lotus. All parts of the American lotus are edible, even for human beings. The petals are edible, the leaves are edible, the rhizomes, which are the stems in the mud at the bottom of the pond, are edible, and the seeds you can actually pop like popcorn, and they're edible. And American lotuses were actually a significant food source for indigenous people throughout this region. But due to the explosion of the muskrat population, almost all of the American lotuses were devoured. Um, Significantly later, because that was about probably what mid to late 1800s. In about the mid 20th century, populations of American lotus started cropping up again all over the Great Lakes region and the northern portion of their range, even though they'd been mostly restricted to the southern portion of their range because of the growth in the muskrat populations in the north. Um, and a lot of people were very surprised by that because many people living in the northern portion of the range didn't know that there were lotuses that were native to that area and certainly had never seen them during their lifetimes. The lotuses were able to survive because, unusually for an aquatic plant, lotuses have a waterproof, not one, but two waterproof or impermeable seed coats. The seeds are heavy, so they sink to the bottom of the pond or lake that they're growing in, and those waterproof seed coats prevent imbibition. They prevent the seed from absorbing any water. So the seeds can sit there dormant for a year, 5 years, 10 years, 100 years without germinating until the seed coat finally rots through both layers, which is a very, very, very slow process. So by the time we're in the mid 20th century, we have lotus seedlings that are actually surviving because the muskrat populations had returned to lower normal levels because their predators had returned. And American lotus were able to survive what could have otherwise been a near extinction by having seeds which have a waterproof seed coat, which ensures that they will sporadically germinate over the course of decades and centuries, which makes their populations extremely resilient. Um, so dormancy can actually be a super big advantage to wild plants, but certainly it can pose some challenges for um, those of us who grow them. Dormancy is caused by many things. It can be caused by seeds which contain chemical or hormonal growth inhibitors. Abscisic acid is a really common plant hormone which promotes seed dormancy. So often we're trying to promote the degradation or breakdown of abscisic acid. Low moisture will also cause dormancy during the desiccation process. Um, Seeds can also be dormant because they might require more light to germinate. So, for example, seeds which require light to germinate, if they don't receive any light, they will remain dormant even if they're provided with moisture um, and optimal temperatures. Sometimes seeds will require darkness. If they're exposed to light, that will induce dormancy. Um, if they're at the wrong temperature, such as lettuce, if lettuce is exposed to temperature, if lettuce seed is exposed to temperature above 80, many of the seeds will go dormant because of high temperature. Sometimes they require scarification. Sometimes a seed will require a stratification, and sometimes seeds require both a scarification and a stratification in order to overcome dormancy. One of the easiest ways to overcome dormancy is pre-soaking. This works for a lot of species. You can kind of get a jump start on getting your seeds germinated by providing them a situation in which they can rapidly absorb a lot of water, whereas they'll absorb water a little bit more slowly if they're directly sown into soil or potting media. Leaching is a process of dissolving or diluting those growth inhibiting chemicals or hormones contained within the seed and washing them away using water. These images that I provided on the right might look like they're not related to this until I explain you how I like to leach seeds. One of my favorite ways to leach seeds is to get a mason jar and some little sachet bags. The sachet bags are made of a kind of loose open mesh, and you can just fill those with the seeds that you would like to leach. You can then place your bags inside of a mason jar. If the seeds are really lightweight and want to float in their bags, you can just get a piece of gravel and put one little piece of gravel inside of each bag to make sure that it sinks to the bottom of the jar. And then what you're going to do is you're going to place your jar under a dripping faucet for a day or a couple days in order to basically have those seeds sit in water. They're going to have a bit of a pre-soak, but also by having the water constantly dripping, water is constantly going to be flowing out of the jar, which means any chemicals which have dissolved out of the seed into the water are able to start flowing away. And after a few days of leaching, often the water in your jar has almost none of those growth inhibiting chemicals or hormones left in it because it's all leaked out of the top of the jar, and your seeds will be capable of germinating. Scarification is extraordinarily valuable for seeds which have an impermeable seed coat. Remember, impermeable means that it's waterproof. And if your seed has a waterproof seed covering, it means that the seed can't absorb water. This is the strategy that American lotuses use. Um, Often what we will do is a mechanical scarification. Um, this individual here in this picture on the right, they're using a file. I prefer to use a piece of sandpaper, and you can basically hold the seed, set your piece of sandpaper on a table, and just rub the seed back and forth really rapidly against that piece of sandpaper until you break through the seed coat. And once you've broken through the seed coat, that seed will be able to absorb water and begin the germination process. Um, Scarification is when we're using acid to basically do the same process of breaking down the seed coat so it can absorb water. Usually we're using sulfuric acid, which is H2SO4. You have to be super, super careful with sulfuric acid because it's a very, very strong acid and it can be very hazardous if not used properly. And you also need to make sure that if you ever do acid scarification, that your acid is diluted to the correct concentration, because if it's too strong, it can literally cook your seed. Um, Heat scarification is the use of heat, such as boiling water, to damage the seed coat. I've done this with the seeds of honey locust trees. My preferred method is to boil water on the stove, and I like to use a very small dish, such as a ramekin, and I put all the seeds that I want to heat scarify in the ramekin. The more seeds, the better. If you don't have very many seeds, they might be overly damaged by the heat. The reason you want to use a ramekin or a small volume of water is because the larger the volume of water is, the longer it will take to cool down, which could mean that you overheat your seeds and you heat damage or kill your embryo. So what I like to do is take my ramekin, pour the boiling hot water that I've just heated up into the ramekin, and then drop all of the seeds that I want to scarify into that boiling hot water. Um, and I'm just going to leave them as that water cools down to room temperature, and that will usually sufficiently damage the seed coat in order to allow them to germinate. Um, and yeah, we've already kind of discussed why, if the seed coat is impermeable, that means that it could take years of seed coat weathering in order for the seed to finally germinate. That could be like seeds in a desert, getting hit by sand grains, blowing in the wind, wearing away the seed coat. It could be, a lotus seed coat rotting at the bottom of a pond. It basically guarantees that they're going to be able to germinate sporadically over the course of many years, which guarantees that even if conditions aren't good one year or the next year or even the next, eventually some of the seeds will germinate at a time where they'll be able to be successful and grow. Stratification is often confused with scarification. Remember, scarification is about scarring or damaging the seed coat. Stratification is not about the seed coat at all. It's about temperature management. Stratification is basically when we are trying to simulate the natural seasonal changes that are required for the seeds of some plants to germinate. Um, Remember that I've just mentioned, plants which have seeds that ripen in the fall, or potentially in the spring. They don't always want their seeds to germinate when the fruits are ripening. So let's take this hawthorn tree as an example. This is the white flowered tree in the top right hand corner. Um, they have seeds which ripen in the fall or winter. But if you plant them immediately, even in a greenhouse, they will not germinate. Um, so they require a treatment or a simulated winter first before they'll be willing to germinate for you. And the reason they would require a winter is because the plant does not want its seeds to germinate immediately. If a robin or a waxwing bird shows up and eats a bunch of fruits off of the hawthorn tree and then poops the seeds out somewhere else, if they germinate in the middle of winter on a kind of maybe a warm, sunny winter day where it gets up to a 60 degrees, if the seed decides, oh, it's good weather, I'm going to germinate. Um, but it's the middle of January, and then the next day it gets down to negative 25, that seedling will die. So the tree doesn't want its seeds to germinate until it knows that they've been through several months of winter, which means now it's springtime and it's safe to germinate. And that seedling is going to be able to grow and mature and develop a woody stem, which is resistant to frost, before the next winter. Um, we can mimic that natural process that occurs in nature with a refrigerator. Um, you got to provide for cold stratification, a very cool condition, cool, moist conditions for several months to simulate a winter. Cool would be defined as 35 to 45 degrees Fahrenheit. 32 degrees is freezing, and at that point, water starts to transition to a solid. And 32 degrees is not a very effective stratification temperature because if the temperature is below freezing, water is solid, and the cells within the seed can't do anything. They can't take up any water. It's hard for metabolic processes to occur because the water is solid. So 35 to 45 degrees is an optimal stratification temperature range, which is easily achievable with most household refrigerators. Um, this is really, really common in most of our temperate woody plants. They often have seeds that require an entire summer to mature. The seeds are shed in the fall. They need to sit in the ground over the winter in moist, cool conditions in order to finish maturing, in order to break dormancy, and then they will germinate in the next spring, where they have the best chance of putting on a lot of growth over the summer to survive the coming winter. Um, some species actually require a double cold stratification. They might require two winters. There are some species of hawthorns that are like this. Some species of hollies also require a double cold stratification, and they might not germinate for 2 years. They might have to go through two winters in order to germinate. But for most species, we're looking at between um, 2 months to 4 months of cold, moist conditions. And so, a really easy way to do this is to collect all your seeds in the fall from trees that you'd like to germinate seeds of, clean them, put them in a nice, fresh, sterile, moist potting mix, not soggy. Put that potting mix in a bag in your fridge and just let them sit for the recommended amount of time for that particular species. Once they've had enough time in the fridge, you can pull them out and then germinate them. Another good way to cold stratify seeds is to build a stratification bed. For example, this bed of sand this raised bed that's full of sand is a great example of a stratification bed. Sand is relatively sterile, so there's less risk of your seeds being killed by pathogens. And you can just sow all the seeds that you would like to stratify in that bed in little rows. And eventually, they will either germinate their first or second year. And if you plant enough of them, often you can kind of get a trickle of seedlings germinating for a couple years if it's a species that requires a couple winters in order to germinate. Some species require worm stratification as well, like some palms. And that normally means 60 to 95 degrees Fahrenheit and moist conditions for several months to simulate a summer. And there are other species that require an even more complex stratification, which we call a warm cold stratification. So they have to be the seeds then have to be subjected to a warm stratification followed by a cold stratification. And a good example of that would be the Helleborus species. We can see a hellebore, which is a perennial in the bottom right-hand corner. Um, and without a period of warmth followed by a period of cold, they won't germinate. Um, if you're not familiar with hellebores, they're one of the best winter blooming perennials. They tend to bloom in late winter before almost anything else has bloomed. One of the names that they've been given is Lenten rose for their tendency to bloom around Christmas time, sorry, around Lent. And in some warmer climates, they might bloom as early as around Christmas. Um, So viability describes the ability of a given seed or seedlot to successfully germinate under the correct conditions, right? So seed is considered viable if once you've provided all the correct treatments and conditions, it is capable of germinating. Um, Viability is one of the most important aspects of seed quality. Viability tends to decline over time, the longer your seed is stored. So it's always a good idea to use fresh seed anytime you want to start a horticultural crop. Um, and the older your seed is, the lower and lower the viability of each individual seed is likely to become, and your entire seed lot or batch of seed is also going to decline. Viability declines due to oxidative damage to DNA. When we talk about oxidative damage, we're talking about um, normal chemical processes that occur within cells that result in what we call reactive oxygen species, which are molecules that contain um, oxygen that is highly reactive. Hydrogen peroxide is probably the most familiar reactive oxygen species. Hydrogen peroxide reacts pretty violently with a lot of different substances, and that's naturally produced inside of cells, and cells have to have ways of getting rid of um, hydrogen peroxide. But in a seed, they don't have great ways of doing that. So if they're not stored properly, they build up oxidative damage to their DNA. They can't repair the damage to the DNA because they're not actively growing. And not just the DNA, but other cellular components like cell membranes can also become damaged under long storage conditions that aren't optimal. Seeds can also be attacked by fungal and bacterial pathogens the longer they're stored. And they're also subjected often to greater degrees of weathering and mechanical damage the longer they're stored. Anytime you like have a container of seeds and all the seeds are shifting around and rubbing on each other, you can experience damage to the seed coat or to the internal portions of the seed, which reduces viability. If you want to maximize the viability of your seed, you want to store your seeds in cool, dry conditions. Unused seed is optimally stored between 35 and 40 degrees Fahrenheit and at a relative humidity of less than 40%. Cool and dry together are going to result in the longest viability period for most seed. And you can see here in this bottom image a, um, a seed refrigerator. This seed refrigerator has the ability to both regulate temperature and humidity within very tight parameters. Some newer household fridges have the ability to um, digitally monitor and also manage humidity. So if you have kind of a fancy household fridge, you can also use that to store any seeds you've collected if you'd like to germinate them later. When we look at the top image, I neglected to mention that this is a viability test. We've got a certain number of seeds here. It looks like we've got 100 seeds here on this little panel of wet paper towel. And what we're doing here is by germinating 100 seeds, we're able to really easily calculate the percentage of viability within this seed lot. And we can see that some of the seeds are well underway with their germination, while other seeds in the exact same conditions have failed to germinate. Um, this is something that seed companies do to guarantee the quality of their seed. Every seed lot is tested to provide a guarantee of seed quality. And often these viability ratings are 80% and higher. It's not unusual to see a rating of 90 or 99% or higher. But just because your seed envelope says that there's a viability of 99% does not mean that it will remain at that viability forever. The longer you store it, the more that viability will decline. So some species of plants have really long-lived seeds. For example, lotus and date palms. Human beings have germinated a couple seeds from plants like lotus or date palms or even a magnolia that are a couple thousand years old, um, and they've still germinated. And we know the age because we can find stored seeds in bottles that were stored by our ancestors in archaeological sites. So we know the age of the seeds because we know the age of the archaeological deposits, ancient cities and whatnot. And then when they're germinated, some species are able to germinate literally after hundreds or thousands of years without any problems. Other species have seeds which lose viability pretty rapidly over the course of maybe, 3 months to 3 years. And good examples would be like African daisies. Some species really don't have seeds which last longer than a single season, like oaks. So if you're trying to germinate oaks, it's always good to collect acorns that current season and plant them almost immediately to prevent loss of viability and seed death. Um, we were just talking about oak seeds, and that's a great example of a recalcitrant seed. We've got two kind of main uh, categories of seeds when it comes to viability. We've got orthodox seeds versus and recalcitrant seeds. Orthodox seeds are the seeds we're most familiar with. They tolerate desiccation super well. They can dry down to less than 10% moisture, often down as low as 5 or 2% moisture, super, super low. Most of these species can be stored for years, sometimes even decades, and we're not going to lose a lot of viability over that time period. I provided two examples of orthodox seeds in the top two images on this slide. Squash seeds and wheat are both examples of orthodox seeds. You can collect those seeds, dry them out, put them in a fridge at 35 to 45 degrees Fahrenheit and 40% relative humidity, and you can keep them for years and years and years and years. I've germinated squash seeds that were 20 years old with no problems before. And that's the ancestral trait of all seed plants. It's obviously very advantageous to have seeds which can tolerate drying out because it allows plants to produce seeds in drier climates. Those soil seeds can sit around in the soil until conditions are moist and right for germination, and then they can sprout. Um, some species, however, have lost the ability to tolerate desiccation. They can't survive it anymore, and if they're going to be germinated successfully, they have to be planted immediately. Um, Good examples would include most of the acorns of oaks, um, citrus seeds, and cacao. Um, Avocados are another good example of a recalcitrant seed. Um, they cannot survive desiccation. They have to be planted immediately. So if you ever want to germinate orange seeds from an orange or maybe from a citrus seeds from a lemon or a lime, you have to pluck them out of the fruit, wash them thoroughly, and then plant them immediately into moist potting media, and then keep them moist um, until they germinate. If they dry out at any point, the seed dies. Same with cacao. If you wanted to grow cacao seedlings, you would have to order a fresh cacao pod or fruit. You would have to extract and clean the seeds of the cacao, and then you would want to sow them immediately after they were cleaned, while they still had a high moisture content, in order to prevent the embryo from dying. Most of our plants with recalcitrant seeds come from moist forests. Why do you think that is? I'll give you a second to think about that. If you thought there's no reason for the seed to ever need to dry out, or even ability for the seed to dry out in a moist forest, that's about right. If your forest floor never dries out, then there's not really a value in drying out ever, because um, you don't live in an environment where that is ever a threat. So you're able to produce a fruit that produces a seed with a lot of moisture in it. That seed is going to land in optimal conditions immediately and be able to start growing. So there's nothing really from an evolutionary perspective that's keeping you from losing that orthodox trait. So just valuable to be aware of. I know a lot of people who have tried to germinate avocados or orange seeds, and they haven't succeeded because they allowed the seeds to dry out before they planted them, and they simply can't tolerate that. So propagation is the idea of making new plants, and sexual propagation is when we make new plants from seeds. Because seeds are the result of sexual reproduction, all of the new plants are at least somewhat genetically unique, even if they're the product of self-pollination. Um, and asexual propagation is making more plants from existing plant parts. So all of the new plants are going to be identical clones. Um, Consider what advantages and disadvantages these modes of propagation might have for a moment. In sexual propagation, because we have genetic variability, we're able to achieve genetic improvement of a crop by removing individuals with inferior performance and favoring plants for future breeding projects that have kind of genetically superior performance traits. Maybe they have larger fruit or sweeter fruit. Maybe they have a larger flower or a new exciting flower color. Um, the problem with sexual propagation is low uniformity. You often have a lot of variability in the quality of your seedlings, which can be a challenge for producers. If you want to produce, an entire field of high quality pumpkins, for example, you want to guarantee that you're using a seed source that is going to breed true and provide a predictable product. Um, in asexual propagation, we can use a superior plant and use cuttings to create a bunch of identical clones, like with this rosemary, um, like these rosemary cuttings. But one of the disadvantages is that it doesn't really allow you to improve on what you currently have. Another risk with asexual propagation is that you often can carry over pests and diseases from the mother plant to the resulting crop. If the mother plant is infected with a virus, for example, all of the resulting cuttings will also be infected with a virus. But with seed propagation, most plants have methods of preventing viruses from infecting the embryos within the seeds. So usually, if you grow from seed, your seedlings will be virus free, which is a big advantage. And finally, percent germination is something that's really important to monitor whenever you're doing germination. That is a percentage of seeds that germinated out of the total number of seeds that were planted. So percent germination, if we look at our this little formula here, percent germination is just your number that germinated out of the total number that were planted. And you're going to multiply that by 100 to get your percent. And most growers are going to monitor this and record their percent germination from year to year for each crop that they grow. Um, and this can be really, really helpful to a grower. Um, how do you think that calculating germination percentages might help you become a better grower? Give you a second to think about that. If you thought that it might help you identify problems with your germination methods, um, that's the biggest advantage. If you get 100% germination one year and then the next year you get 50%, you can analyze what you did differently this year compared to last year and figure out what went wrong, which can guarantee that you always use the best methods of germination in order to make sure that you have a consistent high producing crop. Um, Most commonly, the reasons you've got a low germination rate are you didn't have the optimal germination conditions. So you didn't have the right amount of water or the right temperature or enough oxygen, or you didn't plant at the right depth, or the other problem can be seed viability. If you have a really old batch of seed, or if you have seed that was stored improperly, you'll also have a low germination percentage. So by monitoring your germination percentage, you're able to figure out, oh, did these seeds not germinate because I didn't water them consistently enough while they were sprouting? Did I get a low germination percentage because I'm using the wrong media? Did I get a low germination percentage because I planted them too deep? Did I get a low germination percentage because I've been storing the seed for the last 10 years? These are all things to consider when you are growing any kind of horticultural crop. So I hope you've learned something from this lesson about seeds and germination. And I hope that this helps you in your horticultural endeavors. Have an excellent day.